Greetings everybody. So today we are going to discuss about the classification of the protozoans. So before discussing classification of protozoan, we need to understand the terminology of protozoa, which we have previously had a overview about. So proto is taken from a Latin word proto meaning first, so animals, and protozoans are those animals. These are unicellular animals which had the characteristics of not having a cell wall, had centrioles, and lacked any sort of plastids, as well as they have ingestive heterotrophic mode of nutrition, due to which these are known as protozoans, or because they had animal like characteristics, but since they were unicellular, they were kept in the phylum, they kept in the kingdom Protoctista as phylum Protozoa. Now classification of protozoa in such a manner that here we are having five major classes in which we are going to classify the protozoa. um, of, so here what we are going to do is to classify them five major classes. Number one class is the Sarcodina. Classes includes now the classes which we are going to follow would be according to Morgellus and Schwartz classification. And for that, number one class which is going to be included here would be the Sarcodina. Sarcodina, or in other words, it would be it is also known as Rhizopoda. Class Sarcodina rhizopoda, and that class here involves those protozoans which have do not have a specific locomotory organ, but they do have some locomotory structure, which are the cytoplasmic projections occurring out of their cell, which is known as the pseudopodia. And pseudopodia is among them. And apart from that, they do not have a regular body shape, irregular body shape, as well as the movement which occurs in them is mostly, in most cases, it is amoeboid movement. uh, it is kind of gliding movement occurring due to cytoskeleton contraction, relaxation of cytoskeleton. But uh, as it occurs in an irregular shape, it is known as amoeboid movement. And apart from that, they have a single nucleus. Also, these are free living, some are parasitic as well. And these live in fresh water as well as marine water. Those live in fresh water involves they do not have any shell over them. Examples of these include amoeba proteus, and entamoeba histolytica. And here, entamoeba histolytica basically has amoebic dysentery. It causes amoebic dysentery in humans. And on the other hand side, if we talk about water, marine water marine water Protoctis basically involves the foraminifera, foraminifera, foraminiferas. These are basically living in marine water, and they have a shell which is made up of calcium carbonate, as you may see in the diagram as well. This is a structure of foraminifera. And here they have a shell which is made up of calcium carbonate. Now, they have a reticulate ornament. I mean, this is a complete anatomy, but we are not going through that. And here, these are the structures, as well as there are several pores within their shells. Now we are going to discuss why here. And second type of marine water sarcodine is actinopoda, which is also having a shell, because marine water sarcodines have shell, so they have a shell which is made up of silica. Now coming to why, how is the structure of shell formed? Their shell structure is formed in such a manner that their shell have several spores, and these spores are responsible for the secretion of the cytoplasmic projections, such that they secrete cytoplasmic projections out of these stores, which forms a type of extracellular matrix of thick net, extracellular matrix thick net around their body, due to which they are able to entangle the prey, which in most cases includes bacteria or smaller protists, which they engulf, entangle, right? And uh, here, what we are going to do, is to wait. So here, as you can see, we have the structure. And apart from that, there are some other important sarcodins as well. among which we are having Negoria fulleri, Negoria fulleri, Negoria fulleri, fulleri, and this is basically a member belonging similar to amoeba. It's basically an amoeba which causes primary amoebic dysentery, primary amoebic primary amoebic encephalitis, not dysentery, encephalitis, encephalitis, primary amoebic encephalitis, Negeria fulleri. And apart from that, there's also a very important amoeba, which is a giant amoeba. And giant amoeba, it is the most primitive eukaryotic cell, which has its scientific name as Pelomyxa pelustris. Pelomyxa pelustris is its scientific name, and it basically involves Pelomyxa pelustris. It is a scientific. It is uh, one of the most primitive eukaryotic cell, which generally involves multiple diploid nuclei, as well as it lacks body organelles. I mean cell organelles, and these cell organelles, instead of cell organs, they have engulfed a methanogenic bacteria inside them. Methanogenic bacteria is engulfed, methanogenic bacteria or methanomonas. Methanomonas is engulfed within them. Methanogenic bacteria, methanomonas is methanogenic bacteria is engulfed within them. Right. After that, that methanogenic bacteria is responsible for for providing food, sorry, providing energy to the giant amoeba, and in return the giant amoeba provides it the food for its own survival as well. So in that case, the giant amoeba is benefited by the methanomonas. So instead of engulfing the methanomonas, giant amoeba has kept it as its permanent organelle, and this is a solid proof for the endosymbiotic theory as well. Moreover, when it comes to some other characteristics, giant amoeba here, it lives in below the muddy area of the freshwater, in the depth of the freshwater, wherever there is muddy area, these giant amoeba are present, so that they can get nutrition by the degradation of that organic matter, that muddy area, or from the mud. So in that case, the mud from the muddy area for the degradation of organic matter and the nutritional requirements is taken. Apart from that, when it comes to the other class which I would like to discuss, is about the mastigophora. 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 We also call them as flagellata. Mastigophora. We also call them as flagellata, and these are having a characteristic of flagellates in them. They have a whip-like structure known as flagellate, and they are having a single nucleus once again, as well as they are a single nucleus. Most of these are free living, but some also act as symbionts. Most of these are free living, parasitic as well. Some of these are parasitic as well, and they have some of these are symbionts as well. And apart from that, when it comes to uh, they have flagella for their movement, so these are motile, as well as they have a colorless chloroplast without any sort of chlorophyll. Now here they have got two classifications. Those flagellates which have plastids, those flagellates which have plastids are those flagellates which have plastids and have chlorophyll within them, perform photosynthesis, and these flagellates are known as phytoflagellates. 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 Example of this phytoflagellates includes euglena. Euglena. After that, when it comes to zoo, another example, we are having another class of flagellates, which basically involves all of the heterotrophic flagellates, which do not have plastids inside them, no chlorophyll, no chloroplasts there, which are known as zooflagellates. 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 And examples of these zooflagellates includes flagellates, which includes the conoflagellates, um, trachonympha, trachonympha, conoflagellates, trachonympha are the two ones, trachonympha, conoflagellates, and number third includes uh, trypanosoma, trypanosoma, which causes African sleeping sickness, spread by the vector CC fly, and conoflagellates is a one from where the sponges are evolved. They are from where the sponges are evolved. Now, the structure of the conoflagellates is in such a manner that they are um, colonial flagellates. These are colonial flagellates, conoflagellates. These are colonial flagellates, such that they live in a group of five. They live in a group of five, such that if I'm supposed to draw the structure, let's suppose this is a delicate stalk which is attached to the rock or any other surface, and uh, that stalk basically contains a conoflagellate attached. Let me just try to draw the structure here. The conoflagellate is somehow attached. There are five conoflagellates in a single colony, such that here is a colony of five conoflagellates are arranged. The structure is a bit disrupted, but you can better understand it. And here, as you can see, these are the conoflagellates. Now, these conoflagellates have flagella out of their body, and over that, around surrounding that flagella, there is the presence of um, you know a delicate collar, a delicate collar. And this collar is a characteristic of these conoflagellates because they are having a collar, delicate collar around them, which basically forms the structure, give them the name as conoflagellates. And that conoflagellate are having the same shape, similar shape to conocytes of the sponges. That is why it is deduced that they are evolved from here. Moreover, when it comes to trachonympha are those flagellates which have symbiotic forms, such that these are those zooflagellates which have symbiotic forms with in the gut of termites, such that they cause the digestion of the cellulose present within the gut of termite, and convert it into digestible carbohydrate, which is then sent inside the gut, where the termite are then able to absorb that uh, within their body for nutrition. And apart from that, conoflagellates are generally, if we talk about them, sessile these are sessile zooflagellates, which are in colonial arrangement. Sessile aquatic, both marine and freshwater zooflagellates, which are in colonial form, and they have these form the fawning structure of five. And apart from that, when it comes to trypanosoma, it is a parasitic mode, such that it causes to attack over, it causes African sleeping sickness and is spread by the vector CC fly. Now coming to the next part, which is about the celiophores, ciliata or celiophores class, ciliata, celiophores, celiophores. And these celiophores basically have the characteristic that they have cilia all over their body. They have cilia all over their body for their movement, uh, for the locomotion. And they are having an outer flexible covering known as pellicle, which makes their structure quite flexible and smooth. When it comes to another part which they have a characteristic of having two nuclei. They are having two types of nuclei, among which number one includes one more thing, that the nuclei which they are having can be of same type, can be of same type, they can be of same type, such as monomorphic. They can be of same structure, monomorphic, or they can be of different shape. They can be of same shape, monomorphic. They can be of different shape, dimorphic. So they can have same shape, monomorphic nuclei or dimorphic nuclei as well. Now here, nuclei which are of two types are basically the meganucleus and the micronucleus. Micronuclei uh, lie in number from 1 to 80. In paramecium, we are having only one, and in meganucleus, there's only one meganucleus. And one meganucleus, it is responsible for controlling growth and metabolism, and micronuclei are all involved for sexual reproduction. One more thing that here, sexual reproduction in case of Sarcodina occurs, the asexual reproduction occurs by binary fission, and sexual reproduction occurs by conjugation. Likewise, if we talk about reproduction in Masticophora have their sexual reproduction uh, by means of fusion, as well as asexual reproduction by binary fission. One more thing that the Sarcodina, they are also having sexual reproduction by fusion of gametes. Now you might be wondering how that occurs. That occurs through plasmotomy. Moreover, when it comes to other structure, here we are having their sexual reproduction in asexual reproduction in the paramecium is done by binary fission, and asexual and sexual reproduction occurs by fusion of gametes. These nuclei basically act as gametes because these nuclei are haploid. These are haploid actually. So this is why they are able to perform the function. Now, coming to the other part, they are also having a grooved depression. They have a groove-like structure, a depressed structure within their cell, which is known as the oral groove. And that oral groove basically it is the part where the food is taken in, and that oral groove is basically what we call as which is known as gullet. Oral groove, which is basically here the opening part. Let me just explain the structure over here. So this opening part of that oral groove, this opening part, as we are having in humans, the opening part of the uh, buccal cavity, it is known as mouth. And apart from the space between the lips as well as the teeth is known as, it is an empty space which is known as it is an empty space. This it is an empty space which is known as the vestibule. In that case, oral groove is there. It's a depression. Oral groove is there, which is known as a gullet, and uh, within that oral groove, there is a space which is known as vestibulum. And when the food particles goes through the vestibulum into the cell, it goes through a structure which is known as a cell mouth, or the cytostome, which is known as cytostome. So this is a structure. Moreover, they have anal pores, cytoproct, in order to ingest the digested material, ingest the undigested material. And food vacuoles are also present. As well as for water regulation, they have a special type of organelle known as a contractile vacuole, which regulates the water concentration. And rest there are rest the structure is already defined. Moreover, when it comes to other structures, we are also having other examples of this in Celiospores, such as we are having Paramecium as an example. Moreover, there are organisms which are similar to Celiospores, which is about the next class in which is Suctoria. Suctoria is a class in which we are also having celiospore. The organisms protozoans are also having celiospores in them, cilia in them, but they are only having cilia in their young stage. Cilia in the young stage only, and in the adult stage they do not have any cilia. In adult stage they don't have cilia. They don't have cilia and are um, sessile. Such that at young stage they are motile because they have cilia, and adult stage these are sedentary because they lose their cilia. These are sedentary. These are sedentary, right? Sedentary because they lose their cilia. And apart from that, when it comes to uh, their structure, they are also having two nuclei because that's the reason we are calling them as similar to celiosaurs, because these are having two nuclei. Number one is the meganucleus, and second is the meganucleus, and second is the micronucleus. Moreover, these are also having two. They are having two nuclei, meganucleus, micronucleus. Meganucleus, and apart from that, they are also having some other characteristics, that some of them also have tentacles in order to pierce their prey, in order to catch and paralyze their prey, pierce their prey, and they also have some of them also have adhesive knobs in order to stick to their prey. They have adhesive knobs to stick to their prey as well. A good example of this is Akineta. Akineta. It basically the scientific name is written Podophyra mopasi. Picture is also given that they are having several. As you can see in the diagram, they are having the tentacles. These are the tentacles, and which are utilized in order to uh, pierce the prey. So this is a basic structure. Apart from that, when it comes to our last class, which is the sporozoa, sporozoans, and we also call them as epicomplexans. Sporozoans, epicomplexans, such that these are parasitic. All of these are parasitic, and these are endoparasites. And one more thing, as Protoctist form polyphyletic linkage, the protozoans also form polyphyletic linkage. Parasitic, these are endoparasites, which includes intercellular. They can be intercellular or intracellular parasites, right? And they have a good example of malarial parasite, which is the plasmodium. Moreover, there is an example of a parasite which causes infection in poultry, which is known as coccidiosis, which is caused by organisms of genus Emeria, Ilmeria, Emeria. I'm not sure about the actual spell here. Emeria is a genus of of the class Sporozo epicomplexans, which causes coccidiosis in poultry, as well as uh, we are having they are not having any sort of locomotory organ, don't have locomotory organ or structure, but they do have locomotory organ, have or structure is not present, but they do have um, flexing body movement, flexing body wall movement, which is known as undulating movement, as it occurs in flagella, a wave-like motion, right? So undulating movement, wave-like motion occurs, flexing body wall movement, undulating movement, like with wave-like motion which occurs over here. And after that, when it comes to when it comes to the other part, here we are having uh, they don't have a locomotive organ. Moreover, in order to promote their parasitic mode of life, they spread the infection from one host to another, as well as they complete their life cycle in usually in or more hosts two or more hosts. And apart from that, uh, they have development of special structures known as spores. These spores are responsible to transfer the host. These spores are infective agents which are transferred to the other host. And yes, these are the examples of it. And example of this is Plasmodium. Plasmodium infects organism in such a manner that it is carried by the vector, which is another female Anopheles mosquito. And through its bite, what actually happens is that the plasmodium is released within the blood due to the bite, due to which from the blood it is then transferred to the RBCs, and from the RBCs it is then transferred. From the blood it transfers to the liver cells, where the degradation of RBCs occurs as well as formation of some occurs. So liver cells, and from the liver cells it is then transported to the RBCs, right? And from the RBCs, within the RBCs they multiply, and as they multiply within the RBCs, they multiply to such an extent that the RBC bursts, and as the RBC bursts, it causes more of the plasmodia to come out of those RBCs, as a result of which other RBCs are also infected, until there is characterization of several symptoms such as fever, chills, as well as high temperature, as well as body ache, pain at that moment. And here there is the structure of plasmodium such that it is having a merozoite coat, plasma membrane, pellicular cisterna, as well as it has a rope tree. These are the structural components of it. Polar rings are there, dense granules are there, which are involved in its structure. Meconium, all of these are involved in our structure. That's all about this video. Hopefully we'll meet again in another video.